Okay, well, a very nice introduction was already made about the importance of microbial. We live in a microbial world. We have more or less the same questions behind. Especially microbes allows us to live here in our planet, to adjust temperature, for example, in this model, very old, by the way, but how archaea and bacteria helps to build up the different gases contained in the atmosphere, and also the interaction with the temperature. And so then they make our life, they make us what we are. And we became like microbes hunters, no? From the beginning, and ask, who are they? What are they contributing? How are they contributing? And luckily for us, at least for me, as an oceanographer or something coming from a different discipline, we are in a gold time for having access for microbial communities. And especially because of the molecular view of the living world. And since, of course, all these techniques that are available, that we can use, and especially now that are cheaper to access to this massive sequencing, so we can understand a little more, but we also have more questions, no? Because we have all this increasing richness and of groups that we cannot identify, and of course, then we can ask, okay, what this biodiversity means, and just to show that thanks to these massive sequences, we can access to these rare communities, and as you previously hear, they can be really important also in these different processes in the environment. So what is the perspective that we are using related with this functional meaning is, what this means? And our focus, or my interest, was mainly related with specific or key processes in the environment, especially related with nitrogen cycling. And so then to look for this from natural communities, and then how to study natural communities, their rates, their efficiency, their activities, and in function or in response to changes, physical, chemical changes, and of course in the perspective of all the changes that we are doing and the anthropogenic effects. So the main process that I was interested in was nitrification, which is a very important process in the environment where ammonium can be oxidized all the way to nitrite and nitrate, and of course these are nutrients for other processes, and they can be reduction power, for example, for nitrate in the case of the second step, and they can be also electron acceptors. So then they make the cycle going. And other important thing is that the nitrifiers are also chemoautotrophic groups. So then they have a link between two important cycles, so the carbon and also nitrogen cycle, and a direct role in producing nitrogen gaseous forms, so then participating in nitrogen loss. And what is more interesting now for nitrification, I mean from 2005 on, is that, well, thanks to this massive sequencing, is that we have access to a more diverse assemblage than we used to know. Before it was like two groups main important, but now we know that the groups are related to two domains. For example, for the first functional guild of nitrification and ammonium oxidation, So, for example, for bacteria, we know that there are several genera. Now, ammonium oxidizing archaea, now we know that there are many candidates related to these groups. Also, for nitrate oxidizing bacteria, the second guild that is responsible for nitrification as well. So, there are many, many nitrate oxidizing groups, NOB groups, including the recent potential role of anamox, for example, in these oxygen minimum zones, also as a source of nitrate. So that makes it more interesting to look at which are these functional yields in the environment. And the main interest, or the gradients, or the physical chemical aspects that we are looking for is in marine environments, also in altiplanic salars, because these systems, especially, now in altiplanic salars, you have a natural laboratory of extreme conditions, radiance, salinity, and there is, and in marine environments also. So then, what was our approach was to use two systems, two approaches, two techniques. One related with biogeochemical, making incubations with tracers and also inhibitors of the different processes. And from the molecular point of view, including these massive sequencing strategies, and also evaluating how active they are, how they are contributing using another approach, but following mainly specific groups and genes by qPCR, which has been a very good tool. And then, for example, for the first process, the influence of light. So this is part of our collaboration within the frame of the LOMIC, especially. We have seen, for example, that nitrifying community, this is what we know, there's a very old paradigm related with light inhibition, that the communities, the ammonium oxidizing communities, are inhibited by light. And then nitrate oxidizing communities also. And so this information, this knowledge were obtained by isolates mainly, incubations, and following the rates of these groups. Recently, for example, in the case of archaea, including Nitrosubomylus maritimus, which is one of the most important groups of Taumarchiota, even one of the most important ammonium oxidizers in marine ecosystems, and also in the salars now, there is an influence of how this is the percentage of inhibition, and you can see that compared with bacteria, they are most inhibited compared to bacteria. So, for example, the typical Nitrosomonas, for this case. So then, this is the knowledge that we can get from isolates. But what happened with natural communities? So that was our interest. What happened with natural communities? So what we did was an approach, and this is part also of a PhD thesis of Héctor Levipin, and the contribution of many people here in the lab, Claudia Muñoz, and the staff, Angel. This is the A, I forgot the Rain, the last name. Okay, so, and thanks to the COPAS, we have the availability to go and evaluate daily cycles. So here you can see that this is just the integrated PAR, the radiation. So we can see clearly the differences between summer, verano, and winter time in at least in the intensity, the integrated intensity, light intensity. So then we collect samples during different times along the day, at more than 4 times, and evaluate in the RNA what happened with these nitrifying assemblages along the day, how they respond to light. And we find out that, for example, this is with the activity then with qPCR using functional genes in the case of ammonium oxidizing bacteria, but using phylogenetic markers for nitrospina, which was the main dominant nitrate oxidizing bacteria. And we see that there is a random or different response of the general community, except for the black dots that are Nitrosubhumilus maritimus. We can see that it looks like an inhibition of these natural communities in the environment. But then there is a different response from these other groups. We can see this kind of inverse correlations using this MOA as approach, which is this molecular marker related with ammonium monooxygenase. And also we see this with the pyrosequences, just counting the sequences that we get from Nitrosubhumilus maritimus in the pyrosequencing from all the sequences. And we see the same, but this, for example, for August, that we have this, and also in February, which is our other, we saw exactly the same. So then we can support what have been seen from the isolates, but now in the natural environment. So we can see that there is an inhibition, at least for Nitrosubhumilus maritimus. But then this other more random response of these other communities, what are they doing? What happened then? And then, for example, for the second guild, which is Nitrosospina, as I told you, the nitrate oxidizing, we can see a completely contrasting response during wintertime compared to summertime. So this is open different questions. So in wintertime, we see more or less an enhance of these qPCR numbers in the phenotypes. So of this phenotype, with a tendency of increasing their activities, related with, well, doesn't matter what we write here, actually, we can use the par or it's the same. But during spring, summertime, we see the opposite. So then there must be a threshold in the light intensity which affect this nitrifying assemblage of communities. So then we have a different response. So then we have more questions now. And then for the second, but I will stop for a drink. Sorry. Okay, for the second physical chemical gradient that we were studying, it's related with the response of these assemblages to salinity or substrate. So then we know classical results about the sensitivity of the nitrification processes to salinity. So there is used to be, for example, in estuaries, when looking at the process, there is an inhibition or a decrease of the rate of nitrification in these ecosystems related with salinity. For example, an ecosystem close to your place, France. And then, for example, in other ecosystems, other estuaries as well, from the community's point of view, there is a difference, or it has been shown that there is a difference in the contribution. There is a differential contribution between archaea, ammonium oxidizing archaea, versus bacteria. And this is related with salinity, with a higher contribution of archaea, for example, in low low salinity sites compared to high salinity sites, but also related with the nitrogen available. So then it's hard to exclude both factors. So we have the opportunity to access also with other collaborations, for example, with Claudia Hernandez. So we have a PhD student that is working with us in these questions related with ecosystem services, or in this idea that, so the question of Claudia was to address, well, okay, do this salmon farming have an effect on the communities related with, for example, ammonium enrichment in the ecosystems? So could this have an effect on the communities, on the microbial communities? So we have access to this Comau fjord. I know that it's a very nice place, yeah, but also it's increasing the activity on the Patagonia, but the salmon activities, the salmon farming are increasing in these systems, and so we have access at least for the 3 different periods. One period here where no salmon activity was observed. Here, just look at the spots. There is in these conditions where no salmon activity is the highest amount, or the highest amount of ammonium was related mainly with rivers. For example, the Bodudawe, which is in the head of the fjord. So there's a larger input of ammonium from there, and then you have kind of a decreasing trend. This is just surface. But then, for example, in periods where salmon farming is increasing before, so this is a summer also, this is January, for example, in 2013, we've seen kind of a different pattern. So there's sites where you see a higher surface ammonium concentrations moving or changing towards the sites where the salmon farming is happening. And then, for example, when you have 5 kilograms of salmon in the facilities, for example, close to harvesting time, there is also an increase in these sites, and then the contribution of the river is even less, and this is comparing just to winter times. So what happened with the communities? So there, for example, we had actually we were really lucky because we were allowed to measure in the cages the ammonium concentration. And for example, here is a site for contrasting. You see that most of the ammonium concentration was less than 1 micromolar. But then in the cages, you see very high concentrations of ammonium, around 6 micromolar, which is very, very high for marine environments. Well, I was used to 0.1 micromolar or close to the sediments. You can find, of course, more than, but in the water, surface water is really high. And then what happened with the community? So here we use, we take like these sentinel communities which are really contributing a lot for this nitrification, and then you can see there is a good match between Nitrosubhumilus maritimus, with the ammonium oxidizing bacteria, archaea, sorry, with the nitrate oxidizing bacteria. So they both must be coupled, or our interpretation could be that. So then they are comparable, but then how we can measure in time, how we can really evaluate the activity compared with this time. So then we decided to evaluate not only in the DNA, in this case it was just, this is really potential here, to look at the DNA. But then we evaluate a ratio between the activity found in RNA and DNA. So then it's kind of per cell activity of nitrospina. Just with one of the groups, these techniques are really expensive, so it's not just for quantifying the lab. So then we just focus on one group that we can get a response. And this ratio indicates that the per cell activity, considering that this is a phylogenetic marker, so then the presence is given by the DNA counts, and the activity is given by the RNA counts. So then this ratio allows us to see a trend from no farming activities towards really at the time where the salmons are larger. We can really see a higher activity. So this is preliminary. We're very exciting about this, though, but of course more needs to be done. And then as a second part of this, because this was mainly related with one of the gradients, related with the substrate enrichments, but what happened with the salinity, what I was telling you before, the salinity, and that's what I left to the end, into the altiplanic salars. We've seen that there is a trend in salinity. For example, this is just one of the first campaign. We've seen that this is the main lagoon. All these are salt, accumulated salt. This part is the active, actually the aquatic, the lagoon, the main lagoon. And then there are many ponds around here and the main streams, which are groundwater, that supplies these ecosystems and maintain this evaporitic basin. There's high radiation, high evaporation. So then you have very nice salinity gradients here to explore these questions. So for the first, we've seen a trend from the first campaign towards the other campaigns in the concentration of ammonium, for example, in the main lagoon. The ammonium tends to increase all the times that we went, except for the first campaign. The first campaign was more or less the same, the concentration of ammonium. So then the first campaign allowed us to compare salinity trends only and not the two processes together. And then for the other campaigns, we see an increasing trend also in salinity. So we are evidencing a change in this system. Actually, I visit soon and recently, and it was completely dry. So it really has a very interesting shift during the years. And the salinity in general influenced also the microbial community. So here is pyrosequencing, and we observed that the community structure changed related with these gradients. So there is a notorious, at least in the three pots, that three sites we compare. And here we've done this with considering all the incubations done with biochemical. And we've seen also that when adding ammonium to evaluate the rates, nitrification rates, when we add the spike ammonium, we've seen in the pyrosequencing the appearance of these rare groups. Because what I didn't mention is that most of these groups, nitrifying assemblages, are rare, represent less than 0.1 or 0.4% of the total bacterioplankton community. They are rare. So it's really hard to look at them in a general assays unless using these techniques. Or using a specific, to evaluate a specific with qPCR with specific genes. And by using this, so for example, here you can see, but for the archaea, so here's something complicated with the technique with some of the primers, we can be biased. And then here, these percentages are related with the pyro libraries related only with archaea. Yeah? But if you consider the whole community, then these numbers drops. But we can see that there is a trend also in the groups in the different sites. So, for example, the sites which have a low conductivity sites, for example, less than 3,000, are the ones that have a higher contribution of these assemblages compared to what we observe in the main lagoon in green. Yeah, the main lagoon is also the one that has, here in purple, the lowest numbers of OTUs. So then there is a loss of richness in the high salinity sites, and also in the case of nitrifying assemblages. For example, in the first, I am good in the, yes? Yeah. ¿Cuánto más o menos? No? So then, well, in the first campaign where I told you that ammonium was not a significant it was not significant variable among the different sites, we evaluated the potential nitrification compared to this richness. Entonces, for example, for the site H3, which is the main lagoon, we have a decrease in the nitrification potential. This zone had a low richness, yeah, and have a low richness. So then, compared to these other sites which are would have less conductivity, so then here we can more or less support what we have seen, what have been seen in other environments such as estuaries. So it could be just salinity in this case because there was not a significant trend in ammonium. So then this low richness site has a low nitrification potential as well. But what happened with other periods? So we have the opportunity to evaluate a specific, for example, we decided to evaluate the specific counts for bacteria because bacteria was the main contributor in the case of in functional genes. And then we've seen in these other periods where ammonium, there is a trend in ammonium, a shift in ammonium. Here we have observed that these counts of ammonium oxidizing bacteria were mostly related with the substrate in the water. So they have an increasing trend, and this is the second period. I'm sorry, the gray is the summer, February, well, altiplanic winter actually, February 2012, and then the winter time in 2012. So then we can see that there is a trend related with ammonium available for this community. So there is also a significant relation between this. The activity of the groups are related with the substrate available, which is something that, of course, we can think, everybody can think of, but it's difficult to evaluate if your techniques are really have the sensitivity to evaluate this in the environment from natural community to complex communities. And we could do it from the molecular part. But then also in the case of tracers addition. So using tracers addition, it was also possible to determine an increasing activity, but then here is but mostly for when using one of the inhibitors. This inhibitor, DC7, it's not an inhibitor, it's an antibiotic for archaea. So when we inhibit archaea, then we can evaluate bacteria. The rate was, of course, mostly related with bacteria. And then in this case, we can see a better relations in the green one with the ammonium available. So then the combination of this, so this part of the conclusion, the combination, this approach with using different techniques allows us to explore these natural communities, these key groups, and not only identify them, but to follow them, associated with different physical, chemical gradients. We learned with this that the nitrifying assemblage really responds to these changes, but it's really complex depending on the environment, if there are multiple factors, as in the case of the alkanes or the different substrates available, which is the responsible one. No? So that's what it was helpful for us to go to this extreme environment to evaluate more clearly the response. And of course, in the perspective of the different changes or anthropogenic influence to the environment. So, thank you.